fluorescence. We'll go a little bit into theory and a lot into just how do you move forward when you're trying to design a CLEM experiment for the first time. So briefly, this is all of the work that my lab does. So we study functional protein assemblies. And so we are looking at large arrays of proteins and how those arrays, that ultrastructure, impacts their function. So we do Rubisco inside of carboxysomes, looking at how this polymerization is created, how it functions, why it's there. We do, I used to do some chemotaxis receptor work. So these receptors have to bundle in a certain way, and that allows cooperative activation. Viruses, of course, are incredibly diverse in their applications for different types of lattices. So lattices provide them mechanical strength and also allow them to flip up and fuse with other membranes. And so we work a lot on flaviviruses now. And then finally, lattices are really important to nanotechnology. And so we have a lot of collaborators who are trying to design different machines. And the most important thing is they can always design them really well, but if they're delivering a cargo inside of a cell, they have to fall apart. And that's the hard thing to find sometimes. And so what all of these things have in common is that there is a relationship between the arrangement of the proteins and their function. And so we are trying to always look at both. And this is where the Cryo-CLEM comes in. All right. Pardon me. So question number one, how do I find my target? So this is our standard EM grid. This is, I think, a carboxysome grid. It's kind of hard to tell because they all look the same, right? They're always gray. So your carbon on your grid, the oxygen, the phosphate in the buffer, the nitrogen, oxygen, carbon in your sample, they're all the same elements. And so they tend to look very similar. And so if we're looking for one distinct feature, the question is, how do we know where to point, right? Especially if we're working in a really low focal field, so like a 1 micron field of view. So I have all of these different objects in my grid, and say I'm trying to find a specific one. So this could be a certain endosome inside of a cell where my nanomachine is located. This could be looking for a virus capsid on a grid, an infected cell, large protein cages, anything that is not going to look very different from anything else at this magnification. But the idea is if we can just add a little bit of specificity, we can find our target. And once we make it a little bit different, then we can just point it and we can get going. And so this is the #1 application for Cryo-CLEM, is just tell me where to point the TEM. All right, so these are a couple CLEM applications I pulled from the literature. You will notice this one because it was done right here. And so this is looking at infected cells, and I feel like I should be having somebody else present this. But basically, they're fluorescent if they're infected, and this just tells them where to point, so they can find the cell of interest. Once they get there, they can find their virions, and they don't really need the fluorescence beyond that. It was just really saying, okay, aim here, and it's going to be super bright. This application is a little bit different. This is actually room temperature. So this was done in plastic by Wanda Kolkowski, then a postdoc. Now she has her own group. And what she was doing was tagging specific proteins that are involved in endocytosis. And she used their arrivals to timestamp the order of these proteins and then relate it to the distance inside where the endosome was traveling or the curvature of the membrane at the surface. And so in this case, she was saying, okay, I'm looking at endocytosis, I'm looking at membrane curvatures with the EM, but I need to know which proteins are present, and I can't see that even with subtomogram averaging because they all look alike. And so the fluorescence is giving specificity there. And so what these two applications have in common, obviously they're very different systems, but they both have a ton of fluorophore. And once you can point the TEM, you're good. And so those are really the two features of most Cryo-CLEM applications today. All right, so how this works in practice, I think you've seen all the steps by now. You make a sample, you get it onto a grid somehow, and you throw it into the EM. That's our normal workflow. With fluorescence, we're just adding a fluorophore onto the sample, and we're adding that step of cryolight microscopy. And if you were doing fib milling, it would come right here between the light and the electron. So not adding a whole lot to your workflow, that's the general idea. Now I'm going to show you how we do correlations, and on the left is always going to be fluorescence, on the right is always going to be electrons. This is our handy Leica wide field microscope. That's what we use for all of the fluorescence. I bet everybody in this room has used a wide field microscope before. It operates the same way. The only difference is this little cryo stage and a slightly different objective that can go cold. We can build off of it, and so this is what my lab does. We build different applications. We're working on super resolution right now. And then this is just a standard F20, but you could also use a Krios or whatever you're using. All right, so first things first, we do overviews of both grids. And so this is transmitted light on the left, and you can see the pattern of all the squares really well. And then this is our normal atlas on the right, and you should have seen all that in your practicals, either previous days or today. And what I've circled in blue are features that I can find on both very easily. In this case, I like to use C-flats, and so I tend to break a lot of the carbon. It's a great fiducial. So we can find just two squares and then use that position to orient the grid, and we can find our square of interest, in this case circled in red. From there, we can look at the square. And again, you find bits that are the same in both. And so you can be using something like correlator at this stage. I just use registration markers and CLEM, but you just choose a bunch of things. And so in this case, I'm using empty holes here and here. You could also use chunks of ice. That contamination actually does have a handy purpose sometimes. Cracks, anything that you can see that's similar. And you can use that to do a rough orientation. And this alone will already get you within about 300 nanometers of your target. And for us, most of the time, our field of view is 1 micron, even at high res. So this is enough. And this is really all the correlation that we do at the microscope. So it's very fast. Once we've got this correlated, we can drop in a different color channel. And so in this case, I've registered my fluorescence with my transmitted light. And so now these two are in the same registration. So you can just point to any one of these dots and be taken there on the TEM stage. And so this is one of my correlations. Obviously, it's one of the better ones, which is why I show it here, despite all of this ice contamination. But you can just point and be shown what was fluorescing. In this case, viruses fusing with vesicles. So we're not done. The next part is the post-processing. And so in this case, we're using fiducial markers that are tetraspec beads. So they fluoresce in every single channel. And then the red is labeled vesicles. And so I'm going to identify in both all of the known positions. And so these are my tetraspecs that I can see in the EM, and I can see the green in the fluorescence. And then I'm going to do a transformation matrix, which just minimizes the error between predicted positions. And so you can, again, do this with a number of different programs. I've included a couple down here, but it's essentially the same operation for most of them. The transformations tend to be pretty clean. And then we calculate precision using what's called a leave one out. So in this case, I'm going to leave this one out right here. So I remove it, and then I calculate the expected position. And so I can find by the difference between the actual and the expected what the precision would be, or basically how accurately I'm able to aim for any particular location. And so this kind of post-processing for most applications gets somewhere in the 50 nanometer range. And I think that's kind of a field consensus at this point. People can compete over tens of nanometers, but nobody's going to single nanometer precision. All right, and so this is one of the overviews that you can do at the end, where you can just have that fluorescence overlay, and zooming in, you can see that we've got the vesicles underneath the red cloud, and the, well, you can't really see because it's so bright, but there is a tetraspec underneath the green. So this is what you see in all of your papers. All right, so design strategies. How do I know what fluorophores to use, what applications are going to work? Number one, and this is the most common mistake I ever get when I meet with collaborators for the first time, know your limitations. So resolution. Resolution for these instruments, wide field is about 400 nanometers, confocal is about 250 for the radial, axial or going through Z is god awful no matter what you use. And so just keep that in mind. Remember that it's going to look like a cloud. And so the reason for this is due to the optics of how these systems are set up. And this is a really intractable problem. So this is where a lot of us are trying to work in development right now, getting this to higher resolution. So what's the problem? This would be your normal room temperature setup on the left. You have your sample, cover slip, some sort of oil or water, and then a high numerical aperture objective. It's pretty standard. The oil allows you to get really close to the cover slip. The cover slip is really nice and flat, so you've got an even focal plane, and you can blast your sample with tons of photons, and so get a lot of information out. This on the right is our cryo setup. Now, we've got the objective really far away from the sample, and we have this what's called air coupling. So we can't use an immersion media because cryo temperatures, right? It would freeze. And it turns out air, of course, has a lower refractive index, and so you're going to have lower numerical apertures between the air and this distance. And so what that means is that you're going to collect fewer photons just inherently, like half as many photons if you're lucky. And so that automatically is going to decrease your ability to see. Of course, we're in ice, so this has two problems. One, it's reflective. Okay, three problems. One, it's reflective. Two, it's uneven. So you've got different sample depths across your sample, which will also affect your point spread function or your resolution. And number three, devitrification. So if we blast it with the same amount of light power that we would use at room temperature, we would melt our sample and you would not be able to see anything in the TEM. So we are severely limited in how much light we can use. And this is probably the biggest limitation that we have in cryo. Now, it is dependent on some things. So different metals will transmit heat more efficiently. So gold grids are going to be better than copper. 300 mesh is going to be better than 200 if you're looking just at the devitrification. Ultrafoils are great if you don't want to do tomography on the thin areas. But even with an ultrafoil gold grid, you're still really limited in the power. All right, sample, of course, that's the same no matter what. But then, of course, we have the metal grid. The metal is reflective. You're sticking a bit of metal into a light path. So you've got aberrant reflections going everywhere. We filter them out as best we can, but some still gets through. So what you'll see in your final fluorescence is kind of general low-level dispersed light if you're trying to go for really high gain. And that's just because of all the reflection. All right, so that gives you your functional limitation. And so what this means is I've got a little outer membrane vesicle from Schuanella odensis, and I discovered that there were these little antennae. I have no idea what they are. Nobody else does either. We'd love to know. And I did actually do a mass spec on it, but we got like 800 hits. So say we had an idea of which of those 800 proteins was this guy right here, that beautiful hollow little ball on the end. And the idea with Cryo-CLEM would be, oh, what if I could label it, right? And then I could really know that this is the protein of interest. Well, look at the scale bar down here. So in functionality, this is what I want. This is what I'm going to get. So don't set yourself up for an impossible problem. Remember your resolution limit. I can tell you that the protein is there, but I can't tell you spatially where it is relative to all the others, especially if they all look the same in TEM. All right, precision and accuracy. So again, here's my friendly little Schuanella vesicle. Precision means if I'm throwing darts at a dartboard, how closely are they going to be clustered together? And so it's basically how well can I just group things? Doesn't mean it's accurate, it just means that they're close. And so precision in the correlations is typically around 50 nanometers. So that means that even if I could get the resolution down to beautiful super resolution levels that we're trying to do, they're still going to go like this. And so it's going to be hard to say, is that light coming from the top of that protein? Then there's also accuracy. So this comes mainly from chromatic aberrations, meaning different light colors are going to have slightly different placements on the grid. So it's called chromatic aberration. And we can correct for this. We do this actually with the tetraspec beads because we know that different colors are coming from the same spot. So we can kind of shift things. But this would give you a shift like that. And so this is going to be normal even with corrections. Finally, and this is a really nasty one, most fluorophores do not fluoresce. And so this is because they enter into kind of a non-responsive dark state due to the temperature. And this can be as bad as 85 to 95% for dark. And so instead of getting every single thing labeled, I'm going to get one. And so, of course, then with the resolution, it's going to be that. So the bottom line for all of this is just don't design an impossible experiment. Remember your limitations. We're working on improving all of this, all of us together in the field, but this is the current reality. And so things like throwing a ton of fluorophore onto an endosome, that's great because it's a large object, it's going to be super bright. even if you only see 5% of the fluorophores, you're still going to find it. That's an excellent application. All right. Next thing to think about is consider your sample. So one of the things you have to think about is how bright are the different things in here. So autofluorescence is usually worse. It's because we don't have any photobleaching. That's a great thing with your dyes. It's really terrible when it comes to flavins. So especially your green channel is going to have a lot of punctate autofluorescence. And of course, if you're using tetraspec fiducials, like most of us do, they're super bright. And so I'm generally pushing the limit on how dim things can be because I like to live at the edge. And so what this means is that I'm always worrying about the fact that my tetraspecs might saturate my camera before I can actually see my signal. And so you can deal with this. For example, if you know your signal is going to be dim, you can go into the far red away from the autofluorescence, or you can even order tetraspecs that are missing a certain color, or use single color beads, which will prevent you from correcting from the chromatic aberrations, but it would allow you to see what you're aiming for. So considerations for that. Something else to be aware of is solvent effects, and this one doesn't get a lot of press. So what is a solvent effect? This is a kind of messy diagram that I borrowed from Olympus, but it is trying to show you what's happening with the molecular dipoles. So fluorescence comes from an electronic state. It's just as an S0 to S1. The spin stays the same for the electron. It just flips up to the next energy level. And a ground state, the solvents actually align their dipoles with the fluorophore. And so everything is very nice. When you excite a fluorophore, it basically becomes a different molecule. The molecular dipole gets much larger and it can actually change position a little bit. And so then the solvent over time, while the fluorophore is excited, rearranges to, again, kind of cushion that. It makes the fluorophore a little bit more stable. And you pay for that because a little bit of energy is transferred. And so that's why you get that Stokes shift, that shift towards the redder colors between excitation and emission. And then you'll fall back down. So in cryo, of course, our solvent can't move. And so you're not going to get this effect. So it plays some havoc with the spectra sometimes. But the other thing that can happen is, depending on your different compartments, they have different chemical conditions. And so you're interacting a lot more with the solvent. So this is looking just at ice. So I was basically making the ice more and more crystalline, and I could see that that was having a huge effect on the lifetime of the fluorophore. So what this means is that if you're in the thin edges of your cell versus the nucleus, where maybe your freezing was a little bit slower, that's actually going to affect how bright your fluorophore is. This is another one. The color can change a little bit. So this is just comparing 30% and 50% glycerol. In this case, I'm not thinking so much about the crystallinity of the ice, which is about the same, but I'm looking at the polarity of the solvent. And here you can see there are these big shifts in what colors the fluorophore is responding to. And so what this means is that, again, in the endosome versus in the ER, you have different chemical conditions. Your fluorophore is going to respond to that, and that response is going to be stronger compared to room temperature. And so a lot of interest right now is looking at, the Leica confocal just came out. People are saying, oh, we could do like fluorescence lifetime microscopy. No, not yet, because we don't know what it means. So you could just be simply saying, oh, it's acidic, not, there's this lifetime change that's actually meaningful. So that we have a lot of characterization yet to do to figure out how these interactions are working on a chemical level. All right, and finally, well, not finally, the fluorophore can interact with your carbon. So this is one beautiful example that's awful, which is why I like to show it. So I had a membrane dye, actually. It should have been this beautiful punctate on this grid, but it actually came out of the membrane and onto the carbon. And so your fluorophores may really love the carbon. So this is something you want to screen for. And then, of course, I'm not even getting into the 3D sample preparations. But you can imagine if you've got a large area of your cell that has a lot of depth, you're going to have fluorophores stacked. They might all have different chemical compositions in those respective layers. It can get to be a real mess. So just think through your experiment, know kind of what the most likely failure points are going to be, and either design around it or run some controls and make sure that you really know what you're looking at. All right, and then finally, how do you choose a fluorophore, right? So think about the wavelengths, check your shoulders. So again, these spectra. So this is showing you what light the fluorophore is going to emit at. This is showing you what light it's going to respond to for excitation. Some fluorophores don't respond that much to temperature. And so in this case, you could use the same settings for your filters and you'd be fine. Some of them, there we go. Some of them respond much more strongly. And so this tends to hit the redder fluorophores, which unfortunately are the fluorophores we tend to use to get away from autofluorescence. So a lot of people don't have a setup where they can actually measure this. This is all customized glassware and customized instruments, but just kind of screen through a few filter sets on your instrument and see if you're getting the same picture or if something is suddenly getting a lot brighter or dimmer. All right, color considerations. So of course, I keep saying autofluorescence is going to hit your green channel, so we tend to go red. There's another factor at play. Cameras tend to be more sensitive in the green channel than the red. So if your fluorophore is going to be dim, you're going to have to make a decision between battling with the autofluorescence versus maybe not detecting it as efficiently in the first place. And so one of the things I like to do is I actually will put a labeled sample and an unlabeled sample next to each other in two separate grids, put them both into the microscope, figure out the settings for the labeled one, and then just take a look at what I'm getting for the autofluorescence. And so if your autofluorescence isn't that bad, I would go into the green. If it is bad, go into the red. The brighter, the better. This is always true, but it's especially true with cryo because we're seeing fewer fluorophores. So your synthetic fluorophores are going to have the largest quantum yield, or the amount of fluorophores emitted, of photons emitted versus photons absorbed. Next are going to be your super bright fluorescent proteins. This would be like your M-neon green. That's a great one for cryo. And then finally, the worst performers are the traditional workhorses, so like the EGFP. You can find it, but you need at least 8 EGFP per one visible one. And in reality, most of these systems, you would need about 30 in a point to be able to find it. All right. So the final little bit of my talk, and this is the one that I find the most fun, is my structure functional? So I've done all of this work. I've done my Cryo-CLEM. I found my carboxysome or my virus or whatever I'm looking for. How do I know that this structure is actually the functional form, right? We put all of this work into it, but we don't know that going in. So we can design experiments for this. So this is one that I did during my postdoc, and I was looking at influenza fusion. And this, so I come from fluorescence background from my PhD. And so this up top was what I knew about influenza fusion, because this was all worked out with chemistry. And so you have a glycoprotein that's going to interact with a target. It's going to embed that little green fusion loop, and then it's going to pull down, and you're going to get lipid mixing in the hemifusion state, and then you're going to open up a pore in the fusion. All right, so seems reasonable, right? And you would hope that you could just do a tomogram and see all these states. So the lead group did a beautiful study back in 2016. They actually scooped me, but I don't blame them. But they did exactly this experiment. So they did tomography of influenza fusing. And what they did is they tried to characterize what they saw. So they saw this bridging interaction here, this pinching interaction there, and this extended interface in green. Now, the problem is, how do you relate these two? So we basically have two parallel views of the same object, but we don't have any intersection points. So bridging could be any of the first three areas. The pinching could be pretty much anything on this slide. And the extended interface isn't there at all. So how do we know which of these morphological states are actually the functional pathway towards full fusion? Well, we need to get the chemistry back on the grid. That's the only way to do it. And so I designed a fusion assay to do exactly this. And I'm going to play for you first the tomogram. And I want you to look, you can see actually all of those different interactions on this, bridging, pinching, and extended interface. And I want you to just look at it and see, okay, what do you think is functional fusion? And so to orient your eyes, this fluffy thing, that's your virus filament, and these are the vesicles. All right. So there's a nice extended interface right there. And it's going to come back around. And there's going to be a really nice pinching interaction up in this region coming up. And for everybody who's learning how to do this on a Krios, I did this with a side entry holder. So you can do tomography with lesser microscopes. There's your pinch right there. All right, so what of that is functional? Well, I'm tricking you, none of it. So this fusion assay, the blue would be fusion sites, green are the fiducial markers, and there's no fusion on here. I actually had accidentally created an adhesion arrest condition. And so what this means is that the morphologies really aren't corresponding to the chemistry at all. And so what we did is we designed an assay to be able to look at this more carefully. So we have the fluorescence microscopy, and in this case, the red is the fusion. So it's looking for that hemifusion state or lipid mixing. And then the green is the fiducial markers. And I actually didn't even bother correlating at the microscope. I just did normal TEM, shot everything that looked good to me, because I collect the whole fluorescence anyway. So we've got the fluorescence is generated by this fusion assay. And then I can do the overlays at the end, and I can then relate back. So in this case, this is bright, therefore it must have been fusion. This was dim, therefore it must have been adhesion. And so we can start combining the structure and function into these experiments. So another application that we're working on right now in my lab is trying to do some chemistry in situ. So this is looking at rubisco inside alpha carboxysomes. And we've got concentration on here. Concentration is very easy to measure with the TEM because we know the volume we're looking at. And then S, that's what's called a second order tensor. It's looking at how well everything is aligned. And so if we've got a very low alignment, we have just random orientations, and that you can see in this sparse packing where we also have low concentration. Very high numbers are going to give you this nice ordered, everything is polymerized and along a certain axis. But we also have this third group, the dense, where there's a high concentration, so you'd think it should polymerize, but it doesn't. So what's going on there? Well, we're working on this now, and I don't have an answer for you yet, but I can tell you the workflow that we're developing, and that is with biosensors. So we know the proteins in here are not changing, therefore it must be the small molecules that are, and that are driving this. So we've got our tomography, we're gonna add a biosensor. And so we're going to add a fluorophore that responds to chemical state. Biosensors exist for pH, for redox state, for calcium concentration, a bunch of others. Now, what we have to do is figure out how it responds to cryotemperature, because all of that parameterization needs to be redone for the temperature. But once we figure that out, we're going to be able to do an overlay of the chemical state with the structure. And hopefully that'll give us the answer. If not, it'll at least be a methods paper. All right. And so just to remind you, this is where we currently are. This is what you need to design your experiments for. But this is where we're trying to go. And hopefully in another 5 years, this will be the reality. So with that, I'm going to summarize and thank everybody. Happy to take questions. This actually comprised about 7 years worth of work. It spanned 3 different jobs for me. So I've been in this field for quite a while. And so there are just a ton of people and funding sources that I have to credit. So thank you very much.
Already one question?
Thank you. That was wonderful. One question, you mentioned that the spectral properties of a fluorophore change depending on the state of the eyes.
Yes.
So emission or lifetime, could that be used to check the quality of the eyes on your sample if you know what fluorophore you are looking at?
That's an interesting question. It's actually not one that I thought of. If you had the ability to measure lifetime in place, I think the answer would actually be yes. We would have to make sure to separate it out from the chemical solvent effects that also could affect lifetime. But I think it would work. Yeah.
Thank you.
Hi there, excellent talk. I have several questions, so maybe I'll catch you offline to ask all of them. But one high-level question is, since you're interested so much in methods development, is your lab or really anyone in the room attempting to collaborate with maybe Junelia Farms or even Thermo Fisher to develop fluorophores that are more, well, I guess less temperature sensitive in cryo conditions?
Yeah, so it's a great question. We were briefly partnering with Delmic. The collaboration didn't get off the ground, but just to characterize the fluorophores that are currently available, because developing new fluorophores is great, but especially for people who design fusion proteins inside complex systems, we really don't want to have to tell them, oh, by the way, I know it took you 2 years to label this, but you got to go back and do something else and then do all the controls again. I've actually spoken with a lot of different Cryo-CLEM people, and the number one message I get is, please don't design new fluorophores. Figure out how to make the current ones work. Now, the Myrna group has developed one. I'm actually not sure that they developed it. They may have just characterized it, but PAM-Kate, which is a far red photo-switchable fluor. Photo-switchable fluors usually don't work well in cryo, so that's an unusual one. I'm actually very interested in working with Thermo Fisher because we want to get the resolution improved inside of the fib sum. So we're getting an Aqualos with the IFLM insert, and we actually already have the collaboration set up to try to improve that light path. So yeah, please tell them. I'm happy to collaborate.
So my question is, you're talking about how important it is to select bright fluorophores if possible.
Yeah.
Is photo stability also something to consider or not as much?
That is a great question. So yes and no. So for your normal, like if you have a fluorophore that would photo bleach right away, not going to be a problem in cryo at all, because photo bleaching comes when molecular oxygen collides with the excited state. We don't have collisions. So we don't really get that. What we do see, though, is that all fluorophores basically act like random photo-switchable fluorophores. They're just turning on and off. And the timescale for that is in the tens of milliseconds or even in the seconds regime. So it's very, very slow. We're not quite sure what's causing that. It could be a spin flip of the electron, but it might be something more complex than that. So the answer to your question is something that's designed to be photo-switchable usually won't work well. But all fluorophores will blink on and off, but remain blinking on and off for a pretty long duration. Does that answer your question?
So yeah.
Okay.
Hi, Lauren. We had one question from the online group. How are the performances of Atto dyes and Psi3, Psi5 series under cryo conditions?
Oh,
They're
I'm
trying to determine which to choose between.
I like this person. So the Attos are beautiful. I'm working with the Psi5 right now, and it's also behaving very well. So I think in general, the response is going to end up being that all of the newest generation of optimized synthetic fluorophores are going to be very well behaving. Where we see the weirdness coming in is more with the older generation, like Bodipy, fluorescein, stuff like that, especially Bodipy because it has multiple dipoles.